Plasma membrane integrity is essential for cell function and survival. The architecture and composition of plasma membranes must vary to accommodate the needs of cells within different tissues and based on their specific functions. In this short lecture, we will look at plasma membrane damage and repair mechanisms, followed by an overview of how these different mechanisms relate to disease pathogenesis in those tissues, and we'll review nutritional components that can be used to circumvent that damage or repair it. Plasma membrane damage can occur in response to both chemical disruptions and physical breaches. Let's first look at chemical disruptions. These alter the biophysical properties and lead to breach in a membrane. As an example, oxidative stress and intracellular iron can promote lipid peroxidation of polyunsaturated fatty acids. Damaged portions of the membrane must then be removed, which can destabilize that membrane structure. Membrane lipids are also susceptible to enzymatic damage by both host and foreign lipases. Lastly, alterations in membrane fluidity can occur when amphipathic molecules like alcohol or non-steroidal inflammatory drugs interact with membrane phospholipids or cholesterol is extracted from the bilayer. These fluidity changes weaken membrane structure. Physical breaches can also occur as pictured in this panel. Nanoruptures on the left can be 1 to 10 nanometers wide. These small lesions result from mechanical force or sustained chemical disruptions, while ultimately allowing small molecules to leak out of the cell. Membrane tears are larger, anywhere from 10 to 100 nanometers in size, and result in more extensive leakage of the cytosolic contents. A third type of physical breach is pore formation. This occurs when pore-forming proteins from infectious agents like bacteria assemble into transmembrane pores and alter ion and molecular flux across the membrane. Sources of plasma membrane damage can be organized into 5 major categories. These sources of damage can be highly overlapping, as one type of membrane injury can facilitate or exacerbate another form of damage. Cells experience mechanical stress from physiological events like locomotion, cell migration, and through interactions with inert substances in the local environment. All of these things elicit membrane damage in the form of nanoruptures and tears. Chemical sources include reactive oxygen species present in the extracellular environment or those generated from irradiation that promotes lipid peroxidation. Additional sources of chemical disruptions include amphipathic molecules like NSAIDs and alcohol. These can be comprised of membrane integrity damaging components either through direct interactions or indirectly via oxidative stress. Microbes like viruses, bacteria, and fungi employ a variety of strategies to induce plasma membrane damage. Virulence factors released by these microbes inflict chemical disruptions. They use phospholipase enzymes to do this, and they can also cause physical breaches with the use of those pore-forming toxins. However, there are some larger species that can just exert brute force to damage the host plasma membrane without the use of chemicals or pore-forming toxins. Immune cells can damage cell membranes with pore-forming proteins and antimicrobials. This can occur as part of regular immune functions within our body, including immune surveillance and neutrophil extracellular traps. So far, we've looked at extracellular mechanisms of repair, but there are also intracellular sources as well. Plasma membrane damage can include oxidative stress, which can entice lipid peroxidation and leakage of cytosolic enzymes from lysosomes. Some of these methods are important for cell death and occur through pore-induced mechanisms, but can also be chemical with the use of iron oxidation. Now let's talk about plasma membrane repair pathways. The plasma membrane has many ways to seal a physical breach. This depends on the extent of the damage, and several different pathways often have to cooperatively facilitate wound closure. In this picture, you can see 7 different repair mechanisms, including exocytosis, patching, endocytosis, contraction, plugging, constriction, and scission, which is the cutting out of the damaged portion. Now let's apply these damage and repair mechanisms to 4 different tissues across the body. In this image, you'll see a variety of red arrows. These represent sources of plasma membrane damage, while black arrows denote repair pathways, and gray arrows are indicative of forms of cellular resistance to damage. In the upper left corner, we can see within the stomach the gastric epithelium. Here, the epithelium is highly dependent on having a thick and functional mucus layer, which protects against biochemical and physical stressors. Once that barrier is weakened, this often precedes cellular injury by things like H. pylori pathogen and amphipathic molecules like NSAIDs and alcohol. In the picture, we see physical breaches caused by pore-forming toxins and chemical disruptions, including cholesterol extraction and lipid peroxidation, which are pictured here. Pore formation, cholesterol extraction, and lipid peroxidation can all disrupt the organization of microvilli, and you can see how the microvilli don't look quite like the far left microvilli. They have weakened spots and they curve in different directions that's not quite normal. So how do we repair these things? Well, one method is through lysosomal exocytosis, which we can see here. We also have the ability to use annexin recruitment. Annexins are calcium-dependent membrane-binding proteins, which will bind to the membrane and start to repair and fill in spaces. We also have something called HSP70, which alleviates chemical disruptions, although the role that it plays and mechanism remain unclear. We also have a variety of cytoprotective factors, including calpains and prostaglandin E2. PGE2 stimulates bicarbonate release through a SLC26A9 port, and so then that bicarbonate can neutralize acids that are attacking the cell lining. Below that panel, we have an intestinal epithelium. Here, intestinal cells have to persist in a harsh and dynamic setting, and they can experience damage even at resting states. The 2 primary sources of damage that the intestinal lumen experiences is due to pathogens and ingested chemical stressors like protein carbohydrate complexes or lectins, and also pore-forming toxins that are released by a variety of different microbes that are present naturally in the intestine. When enterocytes experience pore-forming toxin issues, they can get rid of some of those pores through cytoplasmic extrusion. And that extrusion is often initiated through oxidative stress that forms the evidence of lipid droplets, which you can see on the left, and damaged mitochondria. Dietary lectins, which lead to microvilli damage, can inhibit mucus secretions in goblet cells. And that's really an important form of membrane resealing. So when that is not repaired right away, that can become a major issue. The intestine is also a site where autoimmune diseases can further exacerbate these processes, so dietary molecules like polyunsaturated fatty acids or undigested gliadin peptides can promote damage through lipid peroxidation and pyroptosis. This is especially relevant in celiac disease and irritable bowel disorders. Next, let's move to the upper right. Here we see an endothelium. So these endothelial cells line our blood vessels, and they're very important in buffering hemodynamic forces from our heart pumping blood throughout the body. And these tissues, they happen to be exposed to advanced glycation end products, or AGEs. These promote lipid peroxidation and overexpression of receptors for those AGEs can prevent F-actin remodeling, which is required for resealing of damaged cell membranes. On the far right, complement-induced damage can trigger the release of von Willebrand factor, or VWF, and this can limit further complement deposition and damage. So we have a couple ways in which we can circumvent damage through actin remodeling, calveolar enrichment, and then those von Willebrand factors. Lastly, in the bottom right, we have bone tissue, where osteoblasts, the bone building cells, have to work alongside osteocytes, which are mature bone cells. These bone cells, just through regular wear and tear and mechanical stress from us walking around, experience nanoruptures, and these can be repaired through exocytosis, and vitamin E has also been shown to play an important role in limiting further oxidative damage. ATP often leaks from the cells once a wound has been initiated, and calcium-dependent mechanotransduction in nearby locations can help to repair that by signaling P2 receptors in neighboring cells to help begin the repair process. So hopefully you're gaining an appreciation that nutrition has a potential to have impacts across a variety of different tissues. We saw 4 examples, but there are many more. And hopefully you can appreciate that these tissues all had different damage pathways and different repair mechanisms. And that brings up an important point that we have to be careful about balancing treatments and nutritional interventions to make sure that the right tissues are being treated without harming other tissues. So generally, things that may be involved in some of these membrane repairing treatments, those could include things like lipids and sterols, everything from phospholipids, fatty acids, to cholesterol. All of these collectively create the main structure of membranes and add fluidity and rigidity to a variety of different membranes for their function. Minerals and vitamins are important for maintaining oxidative stress levels and getting rid of free radicals. So zinc and copper are important cofactors for superoxide dismutase. Magnesium maintains electrolyte balance through sodium-potassium pumps throughout the entire body, and it also acts as cofactors for other transporters that move substances across membranes. Methyl donors like folate, B12, and choline can promote cell and metabolic regulation through epigenetic DNA methylation. Vitamins C and E act as nutritional antioxidants for cells. Vitamin C acting in watery environments, while vitamin E acts in fat-rich environments. And then vitamin D is an important nuclear regulator of cell expression, gene expression, and it also is a regulator of immune and inflammatory pathways. Lastly, phytonutrients are an ever-growing area of research and clinical benefit. These include plant pigments like beta-carotene, which can act like an antioxidant, and many of these phytonutrients like phytoestrogens, quercetin, resveratrol from grapes, They all have anti-inflammatory signaling properties, which we continue to learn about and have been shown to have benefits for improving membrane integrity.